Hello and welcome to this presentation about new fluorescent proteins. First, I want to thank Sophie Allard and the RTMFM for giving me the chance to make this presentation that I hope you will enjoy. So first, I will describe the variety of the fluorescent protein here, and then I'll turn to new fluorescent indicators per se. Most of the knowledge I have about these subjects comes from my attending the École Physique des Ouches about 2 years ago, and since the last session was cancelled, maybe you'll have a chance to register for the next one by the end of 2020. So first, a bit of history. The Nobel Prize in Chemistry 2008 has been attributed to 3 recipients for the discovery and development of the green fluorescent protein GFP. The first one has purified the GFP protein and shown its basic properties. The second one has shown that it was possible to express the GFP in a heterologous organism such as the worm. And the third one has demonstrated right away that just by mutating the sequence of the GFP, it was possible to change its properties and create new mutants, here a blue mutant for instance. But the GFP those guys worked with, wild-type GFP, is not the one we use ourselves in our labs, because this GFP was dim and was not expressed really well in the mammals. So we had to wait until this work in 1996, which published a list of 30 mutations so that the GFP would accommodate better to transfer RNAs in the mammals and was expressed better and in a more brighter version. Since the golden rule at the time was beta-galactosidase, it was compared to its expression in cells after transfection here, and the enhanced GFP, the eGFP as we know it now, was deemed to be at least as well expressed and as early expressed as beta-gal in those cells. So what is it about the GFP that is delicate to mature? The GFP is actually a barrel of beta sheets, much the same way as DS Red is, although it comes from a different organism, that contains part of its molecule within the barrel itself, and the maturation of which is important because this part gives the fluorescence ability of these fluorescent proteins. This has been deciphered in 2009 by chemists and shown to be long steps. So that's why the wild-type GFP would take 12 hours to maturate, when eGFP would take only 8, and what is called turbo GFP only 2 to 4, although the GFP itself is much the same way as the eGFP. Other interesting properties, such as the quantum yield, may be described in this Jablonski diagram, which shows that a GFP molecule will absorb photons of blue light, and after a short delay will give rise to photons of lesser energy, meaning in this case green light. And that's the cycle we want the GFP to go through as many times as possible. We would like it not to be stuck in any of these states which are non-fluorescent. So the quantum yield is actually the number of green photons emitted per blue photons that has been absorbed by the protein. There's also another property which is interesting, which is photostability. Of course, the quantum yield will lead to the brightness of the protein, but if it becomes extinct very rapidly, that will not work very well. So the eGFP actually displays a higher coefficient, extinction coefficient, which means that it will take more time for the eGFP to become extinct as compared to the wild-type GFP, and that's why it's brighter. Now, a final property I want to talk about is the aggregation of fluorescent proteins, as I experienced myself here in these transfected neurons. DS Red, for instance, will aggregate and make tetramers. So it needed a couple of additional mutations to be monomerized and its spectrum to be slightly slightly modified as well, and it gave rise, for instance, to M. cherry that you all know, and to a whole lot of M. fruits that have been described by the lab of Chen. So, we've shown that there exists a whole variety of fluorescent proteins whose properties are not only the spectra, but also the brightness, photostability, maturation, and lack of aggregation. And what you'll find now when you have publications about new fluorescent protein is proteins with the same spectrum as older versions, like scarlet, which is the new red, but displaying other interesting properties which overall make them brighter. This diagram here shows you the whole spectrum of fluorescent proteins, which is much more diverse than just the EGFP. And now I will focus on this small group of them, which are not really bright, but they've been developed to fulfill a need for fluorescent proteins in the near infrared, because the redder is the better towards imaging within tissues. So it was known for years that a couple of phytochrome photoreceptors of different origin may be able to express fluorescence, and a specific bacterial phytochrome photoreceptor was able to express fluorescence in the near infrared. However, it was considered as bulky because it's composed of 4 different protein domains, and not very practical because it needed a cofactor, biliverdin, which is the product of heme degradation. So for those reasons, it was not really used up until this first publication in 2009, where a version of the photoreceptor was engineered with only 2 domains and the presence of the cofactor, of course, and this way it was able to express fluorescence over 700 nanometers, which was quite impressive. Unfortunately, the fluorescence of this protein depended on either the quantity or the time during which it was exposed to biliverdin. Well, it didn't take long before a new version of it, the IRFP, was demonstrated to be able to be almost as bright in whole organism, this is the whole mice anesthetized, without biliverdin or in the presence of biliverdin. So now we do not have the problem of the cofactor anymore, because every respiring organism expresses heme and then has biliverdin in its circulation. And this IRFP here is sensitive enough to it so that it will always fluoresce when illuminated. After a couple of cycles of mutations and improvements, this last publication in 2019 demonstrated the existence of a nano IRFP, which corresponds to only 1 domain now of the full length that was described before, and at the expense of only a small quantity of brightness, meaning that the nano here is almost as bright as the full length there, as you can see on this scale. Although now it's so small that it is easier to engineer and to functionalize into needs in biology. And we'll turn to that right now. How? When we talk about new fluorescent proteins, we often talk about new fluorescent indicators, and that's when you mutate, or sorry, when you fuse your protein of interest to fluorescent proteins. When you want to do that, you have at least 2 possibilities. Either fuse your protein of interest on the N terminus or the C terminus of the GFP. But about 20 years ago, it was shown that it was possible to artificially circularize the sequence of the GFP and open it at various points, therefore creating new N and C termini and having circularly permuted GFPs, fluorescent proteins. And now you have a full lot of different choices when you want to fuse your protein of interest to the N terminus, to the C terminus, to a regular GFP, to a version of circularly permuted GFP, or even split your protein of interest and fuse each part to a different terminus of a GFP. That's what has been done to produce two families of calcium indicators. One which is the kangaroos that you probably didn't hear of because it didn't work really well, they were pretty dim, and others based on circularly permuted GFPs, the G-Chems, which have been successfully used for the last 20 years. So the G-Chems are a circularly permuted GFP fused on one terminus with calmodulin and on the other terminus with M13 peptide. And the calmodulin displays high affinity for M13 only in the presence of calcium. That means that the structure of the whole protein will differ depending on the concentration of calcium and will modulate the fluorescence of the G-Chem. It is shown on this scheme where when you add an ionophore in the presence of 2 millimolar calcium in the cell, you see the fluorescence of the G-Chem increase in this first version of it. Now, it's been 20 years that this was described, and every year brings a new G-Chem. In April 2020, you can find a new G-Chem FG-Chem 7 here. And it's interesting to notice science has been a huge purveyor of such fluorescent indicators because they need to use them in living neurons to be able to observe their activity. So you'll find plenty of them. I only took a couple, like a voltage sensors here described in 2015, a dopamine sensors, fluorescent sensors in 2018, a norepinephrine fluorescent sensor in 2019, etc. Well, up to now, we've considered fluorescent proteins to be just fluorescent objects, and the quantity of which will depend either on the morphology of the protein itself or the quantity of the protein. But they also display more complex behavior, and that could be useful in biology. So fluorescent proteins may be photoactivated. That means that they are produced in a dark form, and it does not express fluoresce even upon blue light illumination until they are flashed with UV light, which transforms them into a fluorescent form upon blue illumination. It works much the same way for photoconversion, where a green fluorescent protein may be turned into a red fluorescent protein by flashing a UV light that changes the the chemistry of the backbone here. Because there are chemical reactions, these are irreversible. So it could be quite practical to use them to trigger a phenomenon, for instance, going from green to red. It may also, in other times, be interesting to have a reversible reaction, and that's what we have with DROMPA, which is a green fluorescent protein that, upon blue light illumination, gives rise to fluorescence, but also turns to a dark state from which it comes back either naturally or using UV flashes. So it makes cycles. Well, of course, all of these reactions may be seen as drawbacks because usually we'd like the fluorescent proteins to be constantly fluorescent. But some people have been using them, these properties, at their advantage. Here is the classical experiment by the Limpingkott-Schwarz group, where they use photoactivatable M-cherry to make super resolution using photoactivated localization microscopy between the transferrin receptor and PAM-cherry in red and the photoactivated clathrin light chain where they showed it was partially co-expressed. So this is an interesting turnout that made PAM-cherry to rule over PALM for about 10 years, but that may come to an end, because in 2020 a new fluorescent protein was described, the mechanism of which is much similar to that of the near infrared proteins, since it uses a cofactor, bilirubin, and it needs both the illumination and the presence of the cofactor to be fluorescent. So again, that could be seen as a disadvantage But in the case of palm, it means that it's easier to fine-tune the blinking of these fluorescent protein by adjusting the concentration of bilirubin, for instance. And in this very publication, they gave image of palm, which are really striking, of endoplasmic reticulum, vimentin filaments, or clathrin-coated pits. And we'll see in the near future if it holds its promises. Finally, I want to spend the last 2 minutes to talk about optogenetics and how reversible reverse switchable fluorescent proteins may be used in this case. So optogenetics is the fact that in neuroscience again, photosensitive channels have been used to be triggered by light and therefore induce neuronal activity. Well, in cell biology, much the same way, the photosensitive domain, LOVE2, of plant phototropins have been used to mimic naturally occurring inhibitory domains so that upon illumination now, you could trigger the activation of an enzyme. Unfortunately, none of these proteins fluoresce, so if you wanted to follow their expression or see their localization, you needed to fuse them yet again to another fluorescent proteins, which make them bulky. That's why I found this study published 3 years ago very elegant. They harnessed the possibility of DROMPA to go from monomeric and dimeric and fluorescent to lock the kinase, the MEK kinase here, by fusing each extremity of it with one DROMPA. So in the regular configuration here, the two DROMPAs block the catalytic pocket so that this protein here is inactive, and only upon illumination with 500 nanometer light, you'll have the opening of it, and then its activity will be shown. And in this paper, they describe almost undistinguishable reactions, either when you have regular wild-type cells that you treat with serum that activate the endogenous MEK and leads to phosphorylation of ERK, or through the illumination of MEK fusion transfecting in those cells, which also leads to obtain phosphor-ERK. Now, much the same way as other systems, in the baseline is a bit higher with the photoactivatable MEK protein. Anyway, another part that may still be worked on would be the kinetics of the enzyme, since it takes tens of minutes to fully activate, because it needs to translocate out of the nucleus and then be active. In any case, my take-home message is here and will be really simple. There's a whole zoo of fluorescent proteins that it's worth exploring before settling for the good old GFP. And stay on the lookout for the fluorescent indicators you would be mostly interested in, because maybe it's been described in the literature within the last week. Thank you very much for your attention, and stay safe.